When we talk about cancer, the first thing that comes to mind is fear. The fear of something so invasive, so merciless, that it takes control of the very machinery that gives us life. But what if I told you that deep inside every cell there's a hidden weapon that can fight back? A system more intelligent than any drug, more precise than any scalpel, and more powerful than most people realize. It's not a new discovery, it's been with us since the beginning of life. It's our mitochondria. Mitochondria are often referred to as the powerhouses of the cell, but that description barely scratches the surface of their true significance. These tiny organelles are far more than biological batteries. They are dynamic control centers that influence nearly every aspect of cellular life, from energy production to communication, immunity, and even genetic expression. Every heartbeat, every breath, every thought relies on the continuous, precise functioning of mitochondria. They convert nutrients, glucose, fats, and amino acids into ATP, the fundamental energy currency that powers all cellular processes. Yet, beyond this well-known role, mitochondria orchestrate complex biochemical networks that determine whether a cell thrives, adapts, or dies. Mitochondria possess their own DNA, inherited almost entirely from the mother, and this genetic independence reveals their evolutionary origin as ancient, symbiotic organisms. Millions of years ago, primitive cells engulfed these energy-producing bacteria and instead of digesting them, formed a partnership. That alliance became the foundation of multicellular life. Today, that ancient collaboration continues inside us, with mitochondria serving as both energy producers and decision-makers for cellular survival. They sense the cell's internal environment, detect stress signals, and respond rapidly to maintain balance, or what scientists call homeostasis. When cells experience damage, whether from toxins, infections, or mutations, mitochondria are the first to respond. They adjust their metabolic output, shift between energy modes, and if necessary, trigger apoptosis, the natural process of programmed cell death. This is a form of biological wisdom. Mitochondria know when to sustain a cell and when to let it go for the greater good of the organism. It's this delicate balance that keeps our tissues healthy and prevents uncontrolled cell growth. When this communication breaks down, diseases like cancer, neurodegeneration, and metabolic disorders emerge. The unique structure of mitochondria allows them to efficiently handle this responsibility. Their inner membranes are intricately folded into cristae, creating a vast surface area where the electron transport chain operates. This is the site where most ATP is generated, but it's also where reactive oxygen species, ROS, are produced, byproducts, that can act as both signals for adaptation and agents of damage. In moderation, ROS stimulate repair and resilience. In excess, they trigger inflammation and genetic instability. Thus, mitochondrial health is a matter of balance, not just energy. Moreover, mitochondria constantly communicate with the cell nucleus, sending molecular messages that regulate hundreds of genes related to metabolism, stress response, and aging. This crosstalk determines how cells adapt to challenges such as nutrient deprivation, exercise, or environmental toxins. When mitochondria function optimally, they enhance resilience at every level, from muscle endurance to immune defense to mental clarity. When they falter, fatigue, inflammation, and chronic disease often follow. This makes mitochondria not just power producers, but the central command of biological life. They integrate signals from nutrition, sleep, hormones, and emotional stress, converting them into biochemical actions that decide how well or poorly we function. Their role as both defenders and regulators of the cell places them at the very core of our health and longevity, silently shaping every heartbeat and every breath we take. Cancer cells are not random or chaotic invaders. They are master manipulators of cellular machinery, and one of their most cunning strategies lies in how they hijack the mitochondria. Under normal conditions, mitochondria maintain a delicate balance between energy production and cellular signaling, determining when a cell should grow, rest, or die. But in cancer, this balance is deliberately distorted. Cancer cells rewire their mitochondrial functions to create an environment that supports relentless growth, immune evasion, and resistance to treatment. Instead of allowing the natural process of apoptosis, the programmed death of damaged or abnormal cells, these malignant cells suppress it, ensuring their survival even under extreme stress. One of the earliest discoveries in cancer metabolism was the Warburg effect, named after Otto Warburg, who observed that cancer cells prefer glycolysis, a less efficient form of energy production, even when oxygen is plentiful. Normally, healthy cells use oxidative phosphorylation within mitochondria to extract large amounts of energy from glucose. Cancer cells, however, shift to glycolysis, breaking down glucose in the cytoplasm rather than in the mitochondria. This might seem wasteful, but it provides a crucial advantage. It allows them to produce energy rapidly while generating the building blocks needed for cell division: nucleotides, amino acids, and lipids. In essence, cancer reprograms mitochondria not for efficiency, but for speed and survival. But the story doesn't end with energy production. Mitochondria are also the main regulators of cell death, and cancer cells exploit this role ruthlessly. Within healthy cells, mitochondria release cytochrome C when they detect irreparable damage, triggering a cascade that dismantles the cell in a clean, controlled manner. Cancer cells block this process by overexpressing anti-apoptotic proteins such as BCL2 and by altering mitochondrial membrane permeability. This prevents the release of cytochrome C, effectively silencing the cell's natural self-destruct button. The result is a population of cells that should have died but instead persist, mutate, and multiply. Additionally, cancer cells manipulate mitochondrial signaling to resist immune detection. They alter how mitochondria handle reactive oxygen species, ROS, increasing them just enough to promote mutations and tumor progression, but not enough to trigger cell death. They also disrupt mitochondrial communication with the nucleus, changing gene expression to favor metastasis and inflammation. This creates a microenvironment where cancer can thrive, shielded by the very systems meant to protect the body. Another layer of complexity is mitochondrial transfer. Emerging research has shown that cancer cells can even steal healthy mitochondria from nearby cells, rejuvenating their own energy systems and sustaining growth under harsh conditions. This phenomenon underscores how adaptive and resourceful malignant cells can be when it comes to survival. Through these manipulations, cancer transforms the mitochondria from guardians of life into accomplices of disease. By distorting the fundamental processes of energy production, apoptosis, and cellular communication, cancer cells ensure their dominance, turning the body's own survival mechanisms into tools for destruction. At the heart of cancer's destruction lies the failure of one of biology's most elegant defense systems, the mitochondrial self-destruction pathway. In a healthy body, mitochondria are more than energy factories. They are vigilant guardians, constantly scanning for signs of cellular distress. When a cell accumulates too much damage or mutates beyond repair, mitochondria initiate a carefully coordinated process called apoptosis, or programmed cell death. This is not chaos, it is precision. It ensures that damaged cells are quietly dismantled and recycled before they can harm the organism. But in cancer, this natural failsafe is sabotaged. The disease disables the mitochondrial self-destruct sequence, allowing damaged cells to linger, divide, and eventually form tumors. Understanding how to reactivate this mitochondrial pathway has become one of the most promising frontiers in cancer research. Mitochondria control apoptosis through a series of molecular signals involving proteins that regulate membrane permeability. Under normal conditions, these membranes maintain a strict barrier between the mitochondria's inner compartments and the rest of the cell. However, when the cell is under severe stress, caused by DNA mutations, oxidative damage, or metabolic imbalance, apoptotic proteins such as BAX and BAC become activated. These proteins punch holes in the mitochondrial outer membrane, releasing molecules like cytochrome C into the cytoplasm. This release acts as a final signal that the cell cannot be repaired. It triggers a cascade of enzymes called caspases, which dismantle the cell from within in a clean and controlled manner, preventing inflammation or collateral damage. Cancer cells are aware of this mechanism and develop sophisticated ways to suppress it. They overproduce anti-apoptotic proteins such as BCL2, MCL1, and BCLXL, which keep the mitochondrial membrane intact even under severe cellular stress. By preventing the release of cytochrome C, These proteins protect cancer cells from death and allow them to proliferate unchecked. This is one of the main reasons tumors can resist chemotherapy and radiation. These treatments rely on inducing enough damage to activate apoptosis, but when the mitochondrial pathway is silenced, even lethal damage fails to kill the cell. Reactivating this natural death mechanism offers a powerful therapeutic alternative. Scientists are discovering compounds that specifically target these anti-apoptotic defenses, restoring the mitochondria's ability to trigger self-destruction. Some of these compounds mimic the structure of natural pro-apoptotic proteins, tricking cancer cells into opening their mitochondrial membranes and releasing cytochrome C. Others alter the mitochondrial metabolism itself, changing the internal environment in ways that make cancer cells more vulnerable to their own built-in suicide switch. Unlike chemotherapy, which damages both healthy and cancerous tissues, these mitochondrial-targeted strategies are selective. They exploit the cancer cell's dependence on altered metabolism and its fragile balance between survival and destruction. This approach represents a profound shift in perspective. Instead of bombarding the body with external toxins, it turns the body's own intelligence back on. By awakening the mitochondria's natural ability to recognize and eliminate disease cells, researchers are harnessing evolution's most ancient defense system, a mechanism perfected over billions of years to protect life from its own mistakes. The concept of a metabolic switch within the mitochondria represents one of the most fascinating breakthroughs in understanding how cancer can be targeted naturally and selectively. Every cell in the body operates through two main energy systems: oxidative phosphorylation and glycolysis. In normal conditions, mitochondria use oxidative phosphorylation to generate large amounts of ATP efficiently. However, when a cell is under stress or deprived of oxygen, it temporarily shifts toward glycolysis, which provides rapid but less efficient energy. This metabolic flexibility is essential for survival. But in cancer cells, this system becomes permanently hijacked. They lock themselves into a glycolytic state, even when oxygen is abundant, using what is known as the Warburg effect. This allows them to multiply quickly, while avoiding the normal mitochondrial pathways that would otherwise trigger self-destruction. The metabolic switch that reactivates mitochondrial function is essentially the reversal of this hijacking. It involves pushing cancer cells out of their glycolytic comfort zone and forcing them to rely again on oxidative phosphorylation. When this happens, their abnormal metabolism collapses because cancer cells are not equipped to handle the oxidative stress that comes with mitochondrial respiration. The sudden increase in reactive oxygen species overwhelms their defense systems, leading to irreversible damage and triggering apoptosis. Healthy cells, on the other hand, adapt easily to this switch because their mitochondria are flexible and well-regulated. This selectivity is what makes the metabolic reprogramming approach so powerful. It targets cancer's weakness without harming normal tissue. One of the ways this metabolic shift can be triggered is through the regulation of specific enzymes and signaling molecules. Compounds that activate AMP-activated protein kinase, AMPK, for example, can restore mitochondrial energy pathways and inhibit glycolysis. Similarly, substances that suppress the mTOR pathway can slow down cancer cell growth and reactivate the body's natural mitochondrial defense. Nutritional and lifestyle factors, such as fasting, ketogenic diets, and certain plant-derived molecules, have been shown to mimic these effects, pushing cells toward oxidative metabolism and reducing the energy supply available for tumor growth. By manipulating these molecular switches, researchers are effectively retraining the mitochondria to recognize cancerous cells and reactivate their built-in death signals. This metabolic reawakening also enhances the immune system's ability to detect and destroy cancer cells. When mitochondria resume proper oxidative functioning, they improve cellular communication and increase the production of signaling molecules that alert immune cells to danger. Cancer cells, which normally hide from immune surveillance, become more visible and vulnerable. The metabolic switch, therefore, not only disrupts cancer's internal energy system, but also recruits the body's own defense network for a more comprehensive response. The beauty of this approach lies in its precision. It does not depend on external chemicals or radiation to kill cancer cells, but on restoring the natural intelligence of mitochondrial metabolism. By flipping this internal switch, the body's energy systems are recalibrated toward balance and resilience. The mitochondria, once silenced by disease, regain their role as protectors of cellular integrity, dismantling cancer from the inside out through the very process that sustains life itself. Lifestyle, nutrition, and targeted compounds play a profound role in strengthening mitochondrial resilience, which in turn determines how effectively the body can prevent or even combat cancer. Mitochondria are dynamic organelles that respond continuously to environmental inputs: what we eat, how we move, how we rest, and even how we think. Every decision we make influences their performance. When nurtured through proper nutrition and lifestyle choices, mitochondria become efficient, flexible, and capable of neutralizing damage before it becomes disease. But when overwhelmed by poor diet, chronic stress, toxins, or lack of movement, they falter, producing excess oxidative stress, losing energy efficiency, and triggering pathways that favor cellular dysfunction and mutation. This is why mitochondrial health is increasingly recognized as a cornerstone of longevity and disease prevention, particularly in the fight against cancer. Nutrition is one of the most powerful modulators of mitochondrial function. Whole, nutrient-dense foods rich in antioxidants, polyphenols, and essential fatty acids protect mitochondria from oxidative damage and support efficient energy production. Compounds like resveratrol in grapes, sulforaphane in cruciferous vegetables, and curcumin and turmeric activate signaling pathways such as NRF2, which enhance mitochondrial biogenesis, the creation of new, healthy mitochondria. Omega-3 fatty acids from fish and seeds help maintain the fluidity of mitochondrial membranes, ensuring optimal electron transport and energy conversion. At the same time, reducing processed carbohydrates and sugars minimizes the constant flood of glucose that drives inflammation and mitochondrial stress. This metabolic balance allows the body to shift easily between burning glucose and fat for energy, a flexibility that cancer cells lack. Fasting and caloric restriction are additional tools for mitochondrial renewal. During periods of fasting, cells undergo autophagy, a process that clears damaged mitochondria and replaces them with new ones. This not only boosts energy efficiency but also eliminates cellular components that could become cancerous. Ketogenic diets, which emphasize fats over carbohydrates, mimic the metabolic state of fasting by encouraging the body to produce ketones. Ketones provide a cleaner energy source for mitochondria, producing fewer reactive oxygen species than glucose metabolism. Cancer cells, being heavily dependent on glucose, find it difficult to adapt to this shift, which can slow their growth and make them more susceptible to other therapies. Physical activity is another potent regulator of mitochondrial resilience. Exercise stimulates the production of new mitochondria through a process called mitochondrial biogenesis. It also improves oxygen utilization, enhances antioxidant defenses, and balances blood sugar levels, all factors that reduce the metabolic conditions in which cancer thrives. Regular movement essentially trains mitochondria to be more adaptive, more efficient, and more resistant to stress. Targeted compounds and supplements can further support this system. Nutrients like CoQ10, alpha-lipoic acid, magnesium, and acetyl-L-carnitine play essential roles in mitochondrial energy metabolism. Compounds such as PQQ, pyrroloquinoline quinone, and NAD+ precursors like nicotinamide riboside promote mitochondrial regeneration and repair. When combined with lifestyle strategies such as restorative sleep, stress reduction, and exposure to natural sunlight, these interventions create an environment where mitochondria can thrive. Healthy mitochondria are not just vital for preventing cancer, they are the foundation of all physical and mental vitality, influencing how we age, recover, and adapt to the world around us. The vision of a future where natural cellular intelligence surpasses chemotherapy is no longer an abstract dream, but an emerging reality grounded in the science of mitochondrial function. For decades, the dominant approach to cancer treatment is focused on external assaults: chemotherapy, radiation, and surgery, methods that aim to destroy cancer cells faster than they destroy healthy ones. While these treatments have saved countless lives, they also come with immense collateral damage. Chemotherapy indiscriminately targets all fast-dividing cells, often leaving the patient weakened, fatigued, and immunocompromised. What the growing field of mitochondrial medicine proposes is fundamentally different. It seeks not to destroy from the outside, but to empower the body's own systems to restore balance and eliminate disease from within. At the center of this new paradigm is the recognition that the body already possesses extraordinary intelligence at the cellular level. Mitochondria are the executors of that intelligence. They know when a cell is damaged beyond repair, when it must self-destruct, and when it should regenerate. Cancer develops when that intrinsic wisdom is silenced or bypassed. By restoring mitochondrial communication and function, scientists are discovering ways to reawaken this built-in defense, enabling the body to recognize and dismantle malignant cells with the same precision that it once used to prevent them. This represents a shift from a war-based model of medicine, attacking the body to kill cancer, to one of partnership, where the body's natural processes are retrained to restore harmony. Unlike chemotherapy, which creates systemic toxicity, mitochondrial-based therapies are inherently selective. They work by exploiting the fundamental differences between healthy and cancerous cells. Cancer cells depend on a distorted metabolism that prioritizes rapid energy production and suppresses apoptosis. When their mitochondrial pathways are reactivated, this imbalance becomes their downfall. Natural compounds such as berberine, quercetin, and sulforaphane, as well as metabolic strategies like fasting or ketosis, can induce oxidative stress within cancer cells while leaving normal cells unharmed. This precision offers a future where treatments strengthen the body instead of depleting it, where healing enhances vitality rather than diminishes it. Furthermore, the study of mitochondrial dynamics has revealed that their influence extends far beyond individual cells. Mitochondria communicate across tissues through biochemical signals and extracellular vesicles, coordinating responses that affect metabolism, immunity, and inflammation throughout the body. By optimizing mitochondrial health, we are not just targeting a single tumor, but recalibrating the entire biological network that determines resilience and longevity. This systemic intelligence could explain why interventions like exercise, nutrient-dense diets, and stress reduction, often seen as lifestyle measures, show profound effects on cancer outcomes. They do not merely support treatment, they reprogram the body's cellular environment to one that cancer cannot thrive in. As research continues, the promise of mitochondrial medicine points toward a new era of oncology, one where treatment enhances life rather than compromising it. The mitochondria's ability to discern, adapt, and defend reveals that the body already holds the code to its own healing. By understanding and harnessing this code, we move closer to a world where curing cancer may no longer mean destroying the body to save it, but teaching the body to remember how to protect itself.